this is our chapter 13 lecture. We're going to be explaining the molecular basis of DNA replication. Our course objective also includes protein synthesis with that. However, protein synthesis is a different process, and we will talk more about that in the next unit. So we're not going to go in the exact order of the book for this lecture, but I put all the information in this first slide in the order that you'll find it in the book. So we'll go ahead and get started. So if we talk about the levels of DNA structure, we've talked about this somewhat before already, but nucleotides, remember, are the building blocks of DNA or RNA, and then you've got a strand, which is those nucleotides hooked together, and then DNA can form a double helix where two strands are connected to each other and twist in this spirally helical shape. Then a chromosome is the DNA and the proteins that kind of help wind it up, and then the genome is your complete set of genetic material. So all of your genes make up your genome. So if we look at the nucleotides themselves, just like we talked about before, they're composed of a phosphate group, a pentose sugar. That sugar is going to be deoxyribose in DNA or ribose in RNA, because again, DNA's long name, or his name on his driver's license, would be deoxyribonucleic acid, and RNA's long name is ribonucleic acid based on that pentose sugar. And then there's the nitrogenous bases. So the purines are adenine and guanine, and pyrimidines are cytosine, thymine in DNA, and uracil in RNA. So there's a numbering system for the nucleotides. The sugars are numbered on the carbons from 1 to 5. The nucleotide base is attached to the 1 carbon on the sugar. The phosphate group is attached to the 5' carbon, and the hydroxyl group, or the OH group, is attached on the 3' carbon. This is important because we will talk about replication in the 5' end and the 3' end of DNA and RNA. However, I'm not going to ask you to label the carbons on your exam. So if we look at the strand, then the nucleotides are covalently bonded to each other, and in particular we call this bond a phosphodiester bond. So that's when the phosphate groups link the two sugars between the 5' phosphate group and the 3' hydroxyl group. The backbone of the DNA is formed from the sugars and phosphates, so oftentimes we describe it as a sugar-phosphate backbone. And then the bases are going to project away from the backbone. When we write DNA, we write it from the 5' end to the 3' end. So if we were to write DNA strand for this particular picture, we've got thymine, adenine, cytosine, guanine from 5' to 3'. So we would write it TACG. So that brings us to the double helix. Two strands can bind to each other or hybridize if they are both complementary and anti-parallel. So when we say that you have complementary base pairs, that means the A pairs with the T and the C pairs with the G. Chargaff's rule says that any organism has the same amount of adenine as thymine and the same amount of guanine as cytosine because those bases pair in that 1-to-1 ratio. The strands also need to be anti-parallel, so they have to be orientated in opposite directions. So the 5' end of one strand is going to be binding with the 3' end of the other strand. It's kind of like stacking your arms with your fingertips on one side to the elbow on the other side. And when you do that, your other side elbow is going to be with your fingertips. They need to be complementary and they have to be anti-parallel. So what's the term for an organism's complete set of genes? That's the genome. And then a phosphodiesterase bond is a covalent bond specifically between what parts of a nucleotide. That's what makes up that sugar-phosphate backbone. And then provide a complementary anti-parallel strand for this strand of DNA below. So if it's anti-parallel, that one is written 5' to 3'. Our strand would need to be written from 3' to 5'. And then we have CCC, and G pairs with C, so it would be GGG. And then A pairs with T, so we would have AA, and then A would pair with T, so we'd have a T, and then we'd have a G, and then we'd have a C. So it would be GGG, AAT, GC. So bacterial chromosomes, we've talked about this a bit before, but bacterial chromosomes are circular, they're double-stranded, and they have a small amount of protein associated with them. The DNA is going to be supercoiled in a region of the cell called the nucleoid, whereas eukaryotic chromosomes, they're still double-stranded, but they're linear instead of circular, and they are associated with more protein. Chromatin is a complex of DNA and protein, and it's found in the nucleus of eukaryotic cells, and the degree of packing in chromatin actually changes during the course of the eukaryotic cell cycle. So histones are responsible for the first level of DNA packing in chromatin, and a nucleosome consists of DNA wound around a protein core made up of 8 histones. So that's kind of our first level of chromatin packing. So chromosomes are really dynamic in structure, and a condensed region can be loosened or modified as needed for various cell processes. For example, if we think about the structure of a chromosome, most of you are probably thinking about what it looks like in mitosis or meiosis when they are very compacted. The metaphase chromosome is the most compacted chromosome, and it's compacted approximately 10,000 times, whereas the interphase chromatones are compacted typically into a 30 nanometer fiber, which is then folded further in some areas by looping. So we can describe chromatin as either heterochromatin or euchromatin in interphase. So heterochromatin is going to be more condensed, and if it's more condensed, the kind of metabolic machinery that's responsible for getting the information from that chromosome and producing a protein or transcribing genetic information can't access it because it's so tightly wound up. Euchromatin, on the other hand, is more dispersed and it's less compacted, and so that is the kind of chromatin we can actually access and use to make proteins. So that brings us to DNA replication. When we talk about DNA replication, we can say it's a semi-conservative process because each strand of DNA is going to act as a template for building a new strand in DNA replication. So the parent molecule is going to unwind and we form 2 new daughter strands based on the base pairing rules and using the original 2 strands as a template. So at the end, we get 2 new double helices with the same base sequence as the original, and the new double helices have 1 old strand and 1 new strand. So which of the following pictures would represent semi-conservative DNA replication? So that would be the second series of pictures. So we're going to take a little bit of a deeper dive into DNA replication. Copying DNA is pretty remarkable both in the speed that it can happen and the accuracy, and more than a dozen enzymes and other proteins participate in DNA replication, and realistically, we know more about this happening in bacteria than eukaryotic organisms, but the process is pretty similar between prokaryotic and eukaryotic organisms, or at least it is at the level we're going to discuss it. So if we look at starting DNA replication, replication begins at something called an origin of replication, where 2 DNA strands are going to be separated and they're going to form a replication bubble. At each end of the bubble is something called a replication fork, where the parental strands of DNA are going to be unwound. Helicases are the enzymes that untwist the double helix at the replication forks, and then single-strand binding proteins are going to bind to, get this, single-stranded DNA, and that prevents the DNA from recombining. Then we've got something called topoisomerases, which help to relieve the strain that's caused by peeling apart that DNA molecule. If you imagine peeling apart like a rope or some string or yarn, if you peel that apart, it swivels apart and that other end will not, but topoisomerase will break and rejoin the DNA strands to prevent this big ugly knot from happening. So let's go ahead and label the different molecules here. Number 1, who prevents the strand from reforming a double helix? That's going to be C, single strand binding protein. Number 2, who alleviates coiling action caused by helicase? That's going to be B, DNA topoisomerase. And then 3, I mean, they kind of tell you the answer in 2, right? But 3 says travels along 1 DNA strand in the 5' to 3' direction and separates the DNA strands. That's going to be A, DNA helicase. So if we look at the replication in prokaryotic and eukaryotic cells, we said most prokaryotic cells have circular chromosomes, so it's a little bit easier to replicate a circle in some ways. You can see that replication bubble coming out from the origin of replication and having your replication fork there and replication going in 2 directions. In eukaryotic cells, they can actually have multiple origins of replication, and they have these linear strands, and those bubbles spread out and eventually will connect to each other, and you get the two daughter DNA molecules. So if we look at DNA replication and in particular focus on DNA polymerase, it's important to realize that DNA polymerase cannot begin synthesis on a bare template strand. It needs a primer to get started. When I think about a primer, I always think about trying to start my lawnmower. I'm not always super successful, but right, you have to prime the engine before you can get started. So something called DNA primase, or some books call it RNA primase, but either way, DNA primase primes the system here and it gets it ready for DNA polymerase. So DNA primase makes a primer from RNA, and then that allows DNA polymerase to join. So this RNA primer is later going to be removed and replaced with DNA, and these little pieces of DNA that are not quite hooked together totally will be hooked together by something called DNA ligase. So DNA polymerase can only work 5' to 3', and so this kind of gives us a problem as well. The leading strand is okay. It can be synthesized in one long piece, and primase makes a single RNA primer here. The lagging strand, on the other hand, has to be synthesized from 5' to 3', but as Okazaki fragments, and these Okazaki fragments consist of an RNA primer plus DNA. I would strongly encourage you to watch the video I posted in our Canvas site that goes over this. So this image is just showing that DNA polymerase needs to have an RNA primer, and DNA primase provides that primer. So then once that primer is there, DNA polymerase can link nucleotides in that 5' to 3' direction. So here's another picture from our textbook. So primase makes the original RNA primer going from 5' to 3'. That allows DNA polymerase to come in, and this one is making an Okazaki fragment, and then the DNA polymerase is going to detach. And then there's another primer made for another Okazaki fragment, and DNA polymerase 3 comes in again and makes the rest of the DNA. A different DNA polymerase comes in and replaces the RNA with DNA, and then DNA ligase forms the bonds between those two DNA fragments. And again, I would strongly encourage you to watch the video because it's a little bit easier to see in motion. So let's go ahead and match the proteins involved in DNA replication to their function. So who separates the double-stranded DNA molecule into single strands? That is going to be DNA helicase. Who binds to single-stranded DNA and prevents it from reforming a double helix? That is going to be single-strand binding protein. Who removes the titin coil ahead of the replication fork? That's topoisomerase. Which one synthesizes primers? That one's a hard one. That's primase. And who synthesizes DNA in the leading and lagging strands, removes the RNA primers, and can even fill in gaps? That's polymerase. If you look at DNA polymerase and the word polymerase, what's a polymer? Right, many mers. Okay, so a polymer is the large molecule, right? So it's made of many nucleotides. So what's ACE mean? Remember, ACE is the ending for an enzyme. So DNA polymerase is the enzyme that makes the DNA polymer. Looky there. And then who covalently attaches the adjacent Okazaki fragments in the lagging strand? That's going to be DNA ligase. So sometimes DNA can be damaged and sometimes mistakes can be made. You are probably familiar with DNA being damaged with ultraviolet radiation or cigarette smoke or x-rays, things like that. We've actually got something called a nuclease, which can cut and replace damaged stretches of DNA in something called nucleotide excision repair. It's cutting out certain nucleotides and repairing them. There's also several mechanisms for accuracy in DNA replication to prevent these mistakes being made. The hydrogen bonding between complementary pairs is more stable than mismatched combinations. DNA polymerase is also unlikely to form bonds if the pairs are mismatched, and DNA polymerase can even kind of proofread and remove any mismatched pairs. It actually will back up and digest the linkages with any mismatched pairs. So it's not to say mistakes are never made, but we have several systems in order to try and prevent mistakes with DNA replication. The error rate, again, after proofreading for repair is low, but it's not zero. So if we have a sequence change that becomes permanent, and if it can be passed on to the next generation, we get this source of genetic variation, which natural selection can operate on. So a mutation is just a change in a DNA sequence, and mutations can be good, they can be bad, they can be neutral, and they can be nearly neutral. but having variation in a population is something natural selection can work on. And we'll talk more about evolution in Unit 5. So if it's still kind of a mystery at this point, don't worry, we will get it cleared up by Unit 5. Eukaryotic organisms have linear DNA molecules, and so the replication machinery can't complete the 5' end of the daughter strand, and if we keep dividing and dividing and dividing, it's going to produce shorter DNA molecules with uneven ends. However, eukaryotic chromosomal DNA molecules have special nucleotide sequences at their end that are called telomeres. So telomeres are going to be multiple repetitions of one short nucleotide sequence, and they don't entirely prevent the shortening of DNA molecules, but they do postpone it. And it's been proposed that the shortening of the telomere is connecting to aging, because if the chromosomes of a germ cell is a cell that produces sperm or egg. So if these germ cells, chromosomes become shorter in every cell cycle, essential genes could eventually be missing from the gametes they produce. And if these essential genes go missing with aging, that can be a problem as well. However, we have telomerase to come to the rescue here. So telomerase catalyzes the lengthening of the telomeres in germ cells. It increases the length of those telomeres to prevent the shortening of the chromosomes. Really, telomerase is not active in most human somatic cells, so not active in most body cells, but it's inappropriately active in some cancer cells, so it could be a target for cancer therapies. So do you think bacterial cells have telomeres? They don't. Why not? What's the shape of most bacterial chromosomes? They're circular. What's a telomere? It's a region on the end of a chromosome that has repeat sequences. Does a circle have an end? No, it does not. So bacterial cells would not have telomeres. What's a mutation? A mutation is just an alteration of a gene sequence. So when we talk about gene transfer, I think it's easy to think about gene transfer from coming from like parents to offspring or parents to progeny. That's typically what people are thinking of. And that typical pattern of gene transfer is called vertical gene transfer. But that's not the only kind of gene transfer. We can actually have horizontal gene transfer when genes are transferred from one genome to another. Oftentimes you can think about from one species to another with horizontal gene transfer, but it doesn't even have to be from one species to another. It's just from one genome to another, and it's not from parents to progeny in this case. So there's a couple examples of horizontal gene transfer, and we'll talk about this a bit more coming up as well. But transformation is when an organism assimilates DNA into their cells from the outside environment. And we can also have horizontal gene transfer from viruses. In this case, the virus is going to infect cells and take over the cell's metabolic machinery in order to reproduce. And sometimes the DNA from a previous host is transferred into the next cell. So there's an experiment down below pictured that shows you transformation, or a cell picking up DNA from the external environment. So it's a little bit sad, but we've got an experiment where these S cells are injected into a mouse, and the mouse's immune system can't fight it off, and the mouse ends up dying. Then we have R cells that don't have that capsid around them. So these R cells are injected into the mouse can fight those off, so the mouse lives. If we inject heat-killed S cells into the mouse, well, they're heat-killed, so the mouse lives. But this last step is what's kind of fascinating here. So we have heat-killed S cells and live R cells injected into the mouse, and the mouse dies. And when we culture a sample from the mouse, so we grow the bacteria that came from the mouse in a lab, we get live S cells. So that means the R cells picked up the genetic information from the S cells from the environment to produce that capsule. So that is an example of horizontal gene transfer. So now we're going to talk about some lab techniques that are based on understanding DNA structure and replication. I want you to focus on the name of the technique, why it works, and why we would use it, or what the end result is. So we'll talk about each of these kind of coming up here. So nucleic acid hybridization is just when single-stranded DNA or RNA molecules anneal to complementary strands, and realistically this is the foundation of genetic engineering, which is the direct manipulation of genes for practical purposes. So the image that I've got here is where we've got a piece of RNA binding to a complementary piece of DNA, so we temporarily have this DNA-RNA hybrid. So as far as DNA and gene cloning goes, to work directly with specific genes, well-defined segments of DNA and identical copies are going to be prepared in a process that's called DNA cloning. So we're making copies of DNA, and making copies of a particular gene or a single gene is going to be called gene cloning because we're making copies of a gene. Oftentimes, we will use bacterial plasmids and restriction enzymes for this. A plasmid is just a small extra chromosomal piece of DNA from a bacterium. It's like a chromosomal accessory. And then a restriction enzyme is an enzyme that cuts the DNA in a particular sequence of nucleotides. So when we use bacteria for gene cloning, we have to isolate the plasmid from the bacterium. We have to insert the DNA from another source and form a recombinant DNA molecule. If you look at the word recombinant, like recombine, so this is DNA from two sources, the bacterium and then the foreign DNA. This plasmid is then returned to the bacterium, and then what's really exciting, at least exciting to me, is that every time that bacterium reproduces, it reproduces the recombinant plasmid too. And these bacteria can be used to copy this plasmid, or we can even use them to produce the protein product that that plasmid would be coding for, or that recombinant DNA molecule would code for. So the plasmid in this case that carries the cloned DNA is called a cloning vector. So here's the picture that shows you that, right? So we've got a bacterium with a plasmid, which is totally not drawn to scale at all, but they take a gene of interest from a different organism, they combine that with the plasmid, get the bacterium to take the plasmid back up. Every time that bacterium is copied, the plasmid is copied as well. And so you could harvest the protein from the gene of interest. For example, it could be used to produce human growth hormone, or a protein that dissolves blood clots in heart attack therapy. Or you could just use the copies of the gene to introduce the gene for pest resistance into plants, or we could alter bacteria for cleaning up toxic waste and oil spills. So that is how we can use a bacterium to clone a particular gene and what we could do with that. So before we kind of just skimmed over what a restriction enzyme does, but basically restriction enzymes cut DNA at specific spots called restriction sites, and a restriction enzyme usually makes several cuts and it yields restriction fragments. The most useful restriction enzymes cut the DNA in a staggered manner, and they produce something called sticky ends. The ends are called sticky because they can bond with complementary sticky ends of other DNA fragments, and then we can use DNA ligase to close the sugar phosphate backbone of those DNA strands so we can stick together DNA from different sources. So that brings us to another technique called gel electrophoresis. This uses the gel of something called agar to separate molecules of DNA or protein based on the size, length, mass, and electrical charge. These gels are kind of like a Jell-O jiggler. I don't know if you know what those are, but it was like a 90s thing. You would take like 2 packets of Jell-O and put Jell-O on like a cookie sheet and then cut them out into fun shapes so you could like pick up your Jell-O. We'll do this in lab though, so you'll get to see this. And agar is from seaweed rather than bones and hoofs and stuff like Jell-O is. But anyhow, all of that aside, you're going to pipette your sample into these little pores or holes in the gel that are called wells. So you put your sample in one end and then you run an electrical current through, and that separates the molecules out. And so you get these kind of bands. If we look at the bands on gel electrophoresis, the shorter molecules are slowed down much less than the longer or larger molecules, so they move faster through the gel, so they will be farther away from the wells. So that brings us to another laboratory technique called PCR. The PCR stands for polymerase chain reaction. What's polymerase again? So polymerase, remember, is the enzyme that makes the DNA polymer. So this is used to make multiple identical copies of a region of DNA. The cool thing about PCR is that it doesn't need a vector and it doesn't need host cells. So this is artificial DNA replication without any organisms. And after 30 cycles of amplification, a DNA sample will increase 2 to the 30th power. So that's a lot. The key to PCR is that it requires unusual heat-stable DNA polymerases. For example, there's Taq polymerase from bacteria and PFU polymerase from archaea. So these organisms can live in extremely warm environments, and so they produce this heat-stable polymerase in order to do so. And so the reason it has to be heat stable is because with PCR you cycle through denaturation with heat, and then you allow the primers to anneal, and then the new molecules are going to be synthesized, and then you cycle again with denaturing, annealing, and extension, and cycle again with denaturing, annealing, and extension, and so on and so forth there. So PCR amplification alone can't really substitute for gene cloning in cells. However, PCR is great to use to provide specific DNA fragments to be cloned. And PCR primers are synthesized to include a restriction site that matches the site in the cloning vector, and then the fragment in the vector can be cut and ligated or cut and stuck together. And so we get that recombinant DNA plasmid that can be then inserted into a bacterial cell. So DNA sequencing is another technique we'll talk about, and really there's several different techniques that have been developed for DNA sequencing, and it has come a long way here, especially recently. Like, you can take a cheek swab of your dog and get its genetic information. It's nuts. But anyhow, one way we can sequence DNA is by synthesizing the complementary strand of a single immobilized template strand. This technique requires electronic monitors, and they can identify which nucleotide is being added at each step. And if we look at the graph right next to that, you can see that some of the bars are higher on that graph, and that's just showing you that a particular nucleotide has been added an additional time. So there's even more DNA sequencing techniques that are advancing. With nanopore methods, this involves moving a single DNA strand through a tiny pore in a membrane, and the sequence is identified by slight differences in the amount of time the nucleotides interrupt an electrical current across the pore. So I am sure we'll continue to advance DNA sequencing techniques. But the whole point of DNA sequencing is that you get the nucleotide sequence. So CRISPR-Cas9 system is a pretty exciting or newer advancement here. Cas9 is a nuclease. Remember, ACE means it's an enzyme. In this case, it's cutting apart nucleotides. So in particular, it cuts double-stranded DNA molecules. And another exciting thing is that it can be guided by an RNA molecule. So the RNA molecule is complementary to the target gene that we want the Cas9 nuclease to cut. So in the first picture, you've got the Cas9 protein and the guide mRNA, and you get the Cas9 guide RNA complex. This second picture has the Cas9 protein and the guide RNA, and the guide RNA is taken 9 to the particular target gene, and it's cut that target gene. And scientists can use that cut gene to knock out a particular gene, so we can study the normal function of a gene. Like, if you have a model organism that has the gene missing, you can look and see what's missing in that organism's physiology or structure or anatomy. And then we can also repair a mutation in a target gene, so we could cut out a particular area of the target gene. So this CRISPR-Cas9 system, again, can be used to knock out a given gene, or it can be used to repair a gene that has a mutation. This technology is pretty exciting, but there's also some controversy because people get worried when it comes to editing DNA. So let's match the examples to the appropriate technique. So number 9, which technique can be used to remove a gene in a model organism to simulate human disease? So that would be CRISPR. And then what can be used to amplify a few copies of a DNA segment? Really, it should say something more than a few, like a few billion copies. That would be PCR, which would be B. Which technique separates macromolecules and fragments based on size and charge? That's A, gel electrophoresis. And which one determines the order of nucleotides within a DNA molecule? That would be D, DNA sequencing. So in this chapter, we talked about chromosome structure. In particular, we have nucleosomes that are wound around histones. Make sure you know the difference between heterochromatin and euchromatin. Euchromatin is more decondensed or less tightly wound, and so it's actually accessible for producing proteins. We also talked about the structure of DNA, right? You've got nucleotides, you've got the strand, you've got the double helix. We talked about DNA replication, how it's a semi-conservative process. Also make sure you zoom in further in that and can tell me the enzymes and proteins required. Make sure you understand DNA and gene transfer, and electrophoresis, PCR, and CRISPR-Cas9.